Hi guys, wanted to give you a little video on mitosis before we started going over meiosis, just so we can kind of use all that information from mitosis and essentially apply the same exact concepts to meiosis with, I think, one or two exceptions. So I'm just going to write our cell cycle up here. We already went over this, but we're going to divide it into four main areas. We're going to have our M phase. We're going to call this G1, S, and G2. All right. So here is our cell cycle for every normal cell in our body. They're called somatic cells. We have somatic cells, and then we also have sex cells. Sex cells are the sperm and the egg. But just to go over this, for M phase, this right here is when our cell is dividing. So the only cell division happens during M phase. That's its main goal. Its main goal is to take one of your cells, whether it's a heart cell, a lung cell, a liver cell, a brain cell, whatever it might be, and essentially make a copy of it. So we should have two cells at the end of this. After it is done dividing, every cell is going to get its own cell cycle. So it's like essentially the life of the cell. So one of the cells is going to go through G1, and the other cell might be doing the same thing. But it's like kind of like a parent leaving their child alone. They kind of just let them go off and let them create their own life. So anyway, one of the cells is going to go through G1, and all it's doing here is it's growing. Cell's going to get bigger. This is essentially maturing, in your lives, kind of as you guys are growing up, So at this time, all it's doing is getting bigger. The cell is starting to get some of the organelles that it needs to essentially function as its own cell. When it's ready, it's going to go through S phase, and this is where our DNA is going to be synthesized, or we could say it is replicated. All right. So the main thing there is to make a copy of our DNA. And then with the last one, the G2, its cell is going to continue to grow, and it's going to go through some checkpoints. So we'll just call it, say, grows and checks. This is to make sure everything is good inside the cell, the DNA got replicated correctly, so that we can start cell division. And this is a cycle which just keeps going on over and over and over again. Some cells in your body, it happens more frequently than others. Obviously, skin cells are going to be going through this a lot because we're constantly getting new skin. But let's say if it's a brain cell. Brain cells, they just reproduce, and then whenever you're hitting, I don't know, between different people it's different, but we'll say like 22 to 30 years old, your brain cells stop. What we were going to do there for the brain cells is it's still going to divide, it's still going to grow, but whenever a DNA is replicated, that's when we are going to start essentially getting ready to produce a new cell. For a brain cell, whenever we stop dividing them, we don't want to go through DNA replication because we don't want two copies of our DNA. So what's going to happen here is we are going to leave, this is like an alternative route, and we're going to go into G of 0. And what this is essentially the nursing home. The cells are still able to function perfectly. They still live out lives, they still carry out normal functions of cells, but they're no longer going to reproduce. All right, so that's the G0 phase there. All right, this is the main cell cycle, guys. All right, we're going to explore a little bit more of interphase, but we're also going to go into mitosis. For mitosis, we have our four phases. We have our prophase, our metaphase, our anaphase, and our telophase. So prophase, metaphase, anaphase, telophase. And then at the end, we go through something called cytokinesis. So just to go through the pictures of each one individually. Prophase, if this is our cell right here, it's our cell membrane. What's going to happen during prophase is we are going to have a nucleus, and that nucleus starts to disappear. Okay? So inside of the nucleus, we're able to see some chromosomes. And don't forget, we're going to have two copies of each one. I'll just draw two chromosomes here. And then each one got copied, so we have four altogether. So we're able to see our chromosomes. That's prophase, and that's pretty much all that happens. We start to see some centrioles, and they kind of go out to the corners. So there's our centrioles. And that's prophase, guys. That's pretty much it. Metaphase. Okay. We still have our cell membrane. We still have our centrioles there. Only difference here is we are going to take our chromosomes and we are going to line them up in the middle of the cell. Okay? So what's going to happen here, guys, is we are going to have our chromosomes, like I said, lined up in the middle of the cell. But there's one more thing that happens during metaphase. This is where the centrioles come into play. I know we talked about during class the centrioles are like little fishing rods, and they cast out a line to the chromosomes, and they do a particular job with those chromosomes. So here's what happens. Here's our centrioles. They're going to cast out a line to each one of the chromosomes, and these little fishing line things are called spindle fibers. So that is everything that happens during metaphase. You can think of metaphase, begins with an M. Chromosomes line up in the middle. Middle also begins with an M. So that's an easy way to remember that. So right now, guys, our chromosomes are in the middle. Our centrioles have these spindle fibers attached to the chromosome. They also are going to anchor themselves to the cell membrane a little bit. That way, whenever they're reeling it in, the centrioles don't get pulled to the middle. We want them to stay stationary, and we want the chromosomes to separate. All right? So that's prophase, and that is metaphase. Anaphase, I'm going to erase all this. Anaphase, we're still going to have our cell membrane. Everything's normal there. Still going to have our centrioles. But here's what's going to happen. Instead of the chromosomes lining up in the middle, they're going to start to be pulled toward each side. So they're starting to get pulled. In other words, what's happening here, guys, is the centrioles are using those spindle fibers, and just like a fishing line, they're reeling them in. Okay? So we're still connected there. And that's anaphase, guys. All that happens is our chromosomes, which were in the middle, get separated into chromatids, and they go to each side. So our chromatids separate. All right? That is anaphase. Next one, telophase. Here's where we are going to see a pinching inward of our two cells. Our nucleus is going to start to reform. And in each one, we could write the chromatids as separate, or I could put the two chromatids that went to this side together and just make it into a chromosome. Same thing there. Okay, so that's telophase. Again, we have a inward pinching. We start to see two distinct cells, and these two cells should be identical. They should have the same exact chromosomes. Nothing should be different between them. All right. And that's all that happens during telophase, guys. Our last one, cytokinesis, which is essentially, some people put it with telophase as being the same thing, but during cytokinesis, this is when our cells split. Okay, so we did have before, during telophase, we had our two cells there, and we had that inward pinching. Wow, that's a beautiful circle right there, right? We had that inward pinching right there. In animal cells, that's called a cleavage furrow. So what it does is it essentially goes, like this, and then once we get a big enough of an inward pinching, it pulls right apart. Again, for animals, that's a cleavage furrow. For plant cells, it's a little different. It's called a cell plate. So here it's called cleavage furrow. For plants, it's called the cell plate. And this is just an extension of the cell wall, guys. And essentially what happens is it's like a machete, just chops the cell right in half, and we get two new cells. So again, plant cells, animal cells, a little different way. But back to cytokinesis over here, we now have our nucleus back intact. Okay, and we have two distinct cells. This whole process, guys, prophase, metaphase, anaphase, telophase, this is how our cells divide, all right? So the big points on mitosis, guys, that you have to know is when do these events occur during the cell cycle? When is DNA replication? When is it growing? When is it dividing? We talked about the checkpoints in class. We talked about, okay, if a cell is caught by a checkpoint, maybe the DNA has a mutation or something in it. We want to make sure that, hey, that cell doesn't continue to live on. So we just kill that cell, essentially. It's okay, you got millions of other ones inside of your body. But that's what the checkpoints are for. If it gets through a checkpoint, this is when bad things like cancer and other things like that can happen. We do have other mechanisms to make sure we don't get that, but sometimes those mechanisms fail as well. So that's the big deal with the checkpoints. They try to prevent cancer in our bodies. During mitosis, the end results of it, we get two cells, and those cells are going to be identical to one another. That's the big thing. They're going to have the same number of chromosomes. All right. Last thing I wanted to go over with this, guys, is let's say hypothetically, we'll just use humans. All right. For humans, you guys know that we have in our normal cells, we have 46 chromosomes. 46 chromosomes. This is in our normal cells. In our sex cells, we have 23, so we have half that amount. Normal cells that are 46, we call these diploid cells. And if they have 23, we call those haploid cells. Okay? Now let's pretend that we took a normal cell, 46 chromosomes, all right? And we just ended S phase. How many chromosomes would that particular cell have? Well, since we just replicated our DNA, and our DNA has 46 chromosomes, we're gonna have double that amount. So that would be 46 times 2, which is 92. So during S phase, for humans, we would have, at the end of it, 92 chromosomes. That number would continue through G2, where we would still have our 92 chromosomes, but then during mitosis, that number starts to get split in half between the two new cells. So that way our two new cells each will have 46 chromosomes, all right? We'll talk about that 23 number during meiosis, which we'll go over next. But with this, guys, they only have one set of chromosomes. This one has two sets of each of our 23 chromosomes, which is how we get 46. All right? So again, big points to take away, guys, from mitosis is the cells go through one cell division to make two cells, and those two cells are identical to one another. Make sure you guys know all the pictures that I gave you for mitosis, and I think that's pretty much it, guys. Hopefully this video helped. Have a good night.